Alrighty, hi everyone. My name is Adam Moore, and this is another iteration of the People's CDC Grand Rounds. And today I'm going to talk to you all about SARS-CoV-2 variants. And today's talk is a bit more biology heavy than the last one, but I am hoping I'm able to break everything down simply enough to where we can all understand it. A lot of this kind of echoes back to some high school biology, so maybe some will be familiar to you all. So let's dive in. Um, First, I want to talk about genomes. So these are either made up of DNA or RNA, and a genome is the entire genetic material of a living being. It's the collection of all of your genes. So for DNA, it looks something like this. It's a double-stranded helix. So we often think of it like a spiral staircase. RNA is essentially that, but cut in half vertically. It's just single-stranded. So these are DNA and RNA. When I reference those, this is what I'm referring to. And to understand how variants work, we need to understand how genomes are replicated, how copies are made inside the body. And so to understand that, we need to know what an enzyme is, which simply is just a protein that helps make a chemical reaction happen. And these are crucial for genome replication. So a kind of simplified version of that here on the bottom blue bar is a genome. And then on the top is the copy that's being made. And then the purple oval is a genome copying enzyme. So it goes along the genome, reads it, and makes a copy of it like this. Now, sometimes errors happen, and this is when mutations come up. And when you're dealing with DNA, if there is an error, when DNA is being replicated, there is a proofreading mechanism built into these organisms. And so a proofreading enzyme can come on by, sees that there's an error, and fixes it, and then the replication continues. But if you're dealing with RNA, there is no proofreading mechanism. So that mutation, that error stays in there and it's never fixed. That's now part of the genome copy that has been made. And so this is, of course, a central part of a virus's life cycle. Um, I should say that every replication is an opportunity for mutations, whether that's DNA or RNA. Every time a genome is copied, there is a chance for error to occur. And this is very much the case when we think about viruses. And so I'm going to show you a simplified diagram of how a virus can infect and replicate inside of a cell. So here, starting at number 1, we have a virus binding to a receptor on a cell, and that allows it to enter the cell. And then step 2, excuse me, the virus is spitting out its genome into the cell, and then the cell starts to make copies of this genome. And then from there, a new virus can be assembled, and then the cell spits it out. And sometimes the virus kills the cell when it's released, sometimes it doesn't. This is very dependent on the specific virus in the specific cell. But in general, this is how the process works. You bind and enter the cell, you replicate the genome, like I just showed you in the previous slide, you make new viruses, and then they're released. And that's a simple virus life cycle. But we're not talking about all viruses today. We're talking about coronaviruses specifically. So this is a family of viruses, and some of the more famous ones are, of course, SARS-CoV-1 and 2. SARS-CoV-2 being the virus that causes COVID-19, MERS, which is Middle East Respiratory Syndrome, and then some common colds are caused by various coronaviruses. All of the viruses in this family are RNA viruses, which means they're naturally prone to mutating because they don't have that proofreading mechanism that DNA-based organisms have. And for coronavirus genomes, there are two major components. There are genes that make structural proteins. So these are literally the structure of the virus, the spike proteins on the outside, the envelope that kind of coats the entire virus. And then there are genes that make non-structural proteins. So these are like the enzymes that help make copies of the genome. But these are the two major simple components of the genome, structural and non-structural. And now, of course, we're here to talk about variants. So that means we're talking about mutations in which are very common in these coronaviruses. In The impacts of mutations are not always straightforward. Often mutations don't have any impact at all. Just because a mutation is present doesn't mean it's actually going to do anything. But they also can very much have impacts on the virus. They can have structural impacts, so changing the shapes of proteins on the outside of the virus, which can impact how a virus binds to a cell. It can make it quicker, it can make it slower. How a virus binds to an antibody, which can make the antibody more or less effective. Um, and this can also, non-structural mutations can impact how quickly or slowly the virus replicates if there are changes to the enzymes that the virus genome is making. And so for the remainder of this talk, I'm going to talk about variants just in the scope of Delta and Omicron, just for the sake of simplicity. What I'm showing you isn't necessarily, this definitely happened with Delta and Omicron, but these are just two variants that we all know the names of. So I thought they would be an easy example for us to follow. So let's say the Delta variant looks like this. These are the spike proteins on the outside. They have all these points. And the Omicron variant looks like this. The spike proteins are pretty similar, but they have some noticeable differences where some of the spikes are now rounded. And so the proteins on the outside of Omicron are different than Delta. But how do this is a simple example of some structural impacts of mutations. With a different variant, the structure is looking a little differently. But how do these variants actually come up? Well, technically, variants for COVID, for SARS-CoV-2, are classified when there is a functional change that has occurred. And this criteria is pretty subjective. There's a committee of people working with the WHO in various countries that are analyzing genomes, running experiments to see how things may or may not have changed, and that helps determine how to classify and name a new variant. So we have this system here where we have a genome copying an enzyme copying the SARS-CoV-2 genome, a mutation pops up. But like I said, one mutation doesn't necessarily mean there's going to be an impact. But if you get enough mutations, you get a lot of them, your chances of having a functional impact increase. So if you get enough mutations, you can get a new variant. And because there's no proofreading mechanism, these mutations are cumulative. They just keep adding on and on. And so that's one way a variant can come up, is just more and more mutations keep happening. Another way variants come up is through virus recombination. And the coronaviruses are very famous for doing this. They're not the only viruses that do it, but they're known to do it a lot. And so recombination is essentially when a new variant is made by two or more existing variant genomes mixing together. And this happens when someone is infected with multiple variants. Maybe they're exposed to more than one variant, or maybe they have a chronic infection with one variant and then they're exposed to another variant. And the way this recombination happens is, let's say someone has Delta and Omicron, and an enzyme is making a copy of the Delta genome. What coronaviruses can do is essentially some of the Delta genome is copied, and then the enzyme just hops over to the Omicron genome and starts making a copy of that and just tacks it onto the end of the Delta copy. So now you have this new variant as a result. You have a hybrid of Delta and Omicron put together. So that is another source of potential variants, is this recombination. And one big worry that always comes up with new variants is, how is our immunity going to hold up? How do we make a new vaccine that is going to address this new variant? And when it comes to SARS-2, we are often talking about immunity in terms of antibodies. This is not the only important branch of the immune system when it comes to fighting off a virus, but it's what we talk about a lot with SARS-2. So I want to talk to you all about how antibodies work. And this symbol up here is just a basic structure of an antibody. So there's two major ways that antibodies work with viruses. First is neutralization, where you get enough coverage around a virus so that it cannot attach to a cell. It can't invade, it can't replicate, it's neutralized. And so when this happens, your body just clears it like normal, any sort of detox that happens in your bloodstream. The other way that antibodies work is through a process called opsonization, which in a very simplified way is you get maybe a few antibodies covering the virus, and then these antibodies are signaling to other cells in your immune system to come eat it. So they're like, hey, dinner is served. We got a nice meal for you. Why don't you come on in, eat this virus, and that will destroy it. And that's happening by other immune cells, a variety of them. Um, and so, with these two major methods of how antibodies work, it's important to, especially in the face of variants, it's important to understand how antibody binding happens. So, antibodies are always unique to a given virus. You know, a measles antibody is going to be pretty useless against SARS-CoV-2. And antibodies are actually optimized over time through a process called antibody affinity maturation. And so when you are first infected with a virus, your body is going to pump out some antibodies and they're going to be okay, but they're not going to be the best that they can be for a virus. Over time, even after the infection has ended, your body is continuing to make the antibodies a better and better match to the virus that it just saw. So it can bind to it better. The affinity has matured, antibody affinity maturation. And again, this helps make the antibodies more effective for the next exposure. It's part of your immune memory system, and it's how we just get this process optimized to where if and when you are exposed again, you are in a really good position to fight it off a lot quicker. And these optimized antibodies can still work to a degree against a different variant. It's just the effectiveness is very dependent on the next variant you're exposed to. So antibodies against Delta can still be somewhat effective against Omicron, but there are some important concerns when it comes to an antibody against a variant like Delta working against a variant like Omicron. And again, these are just general examples. Um, so for this visualization with antibody binding, we have a Delta spike protein and it's coming into contact with an antibody made for Delta. That's a pretty solid fit. But now we have an Omicron spike protein coming into contact with a Delta antibody. And that fit isn't quite as good. That binding isn't as strong as the Delta antibody and Delta spike protein. And so when you have non-optimal antibodies, there's a chance for poor binding, and the antibody may not even actually stay attached to that virus, which means that virus can still invade your cells and make copies and continue making you sick and causing damage. And so, getting these updated antibodies to more accurately and more effectively bind to a variant you might be exposed to is a big benefit of getting booster shots. Booster shots help create another set of antibodies that are a much better match to existing variants before you are actually exposed. This is essentially kickstarting your antibody affinity maturation process before you actually get a real life exposure that's going to make you sick and cause some damage internally. And this can still happen after exposure, but of course, once you're exposed, then you're actually getting sick. If it's happening beforehand, your body is essentially getting the booster and saying, okay, this Omicron spike protein that's in this booster, that's pretty similar to the Delta that I saw, but it's not quite the same. So let me make a new set of antibodies that are a better match here. And that makes your immune system better primed for how the virus has changed. And one way you can think about this is like getting plastic surgery on your face and Face ID on your phone. If you have too much surgery without updating your Face ID, your phone isn't going to recognize you. If too many variants go by without you getting a booster, your immune system isn't as prepared. The virus has undergone too much plastic surgery for your antibodies to recognize it as well as they could have if you had gotten a booster. So another way we can think about this is looking at variant progression with a phylogenetic tree, which is just the scientific way of saying a family tree based on genetics. And it looks something like this, where we have the origin on the left with our virus here. We can just call this variant A. And then as the virus evolves, the tree expands, and each of these branches are a variant, and then more variants coming off, and so on and so forth. So let's say this is our variant phylogenetic tree, and right now variants J and K are circulating. But your last shot was for variant A. Now, having a shot for variant A is much better than nothing. However, excuse me, a lot has changed with this virus to get all the way to J and K. Your antibodies for variant A can still work to a degree for J and K, but there's a lot of affinity maturation that has not happened. There is a lot of changes that your body has not been primed for. So if you get a booster for B, you're even closer to the more current variants. That's better. In D and F and so on, the closer you get, the more prepared you are. And so these boosters are helpful even if they are for earlier variants. If you get exposed to J and your last booster was for D, that's still a lot better than your booster for B or A because you are getting closer and closer to the current variants. And so when booster shots are coming out and they're designed for a variant that is no longer the major variant that's circulating, maybe it's not circulating at all, the current variants that are circulating, you have a pretty good bet that they are going to be descendants of the variant that inspired the booster shot, which means those booster shots are still going to be a fantastic tool for priming your immune system to make antibodies that are closer and closer to the variants that you're exposed to. So to bring it back to this visualization, we have delta spike protein binding to a delta antibody. It's a great fit. But then you got an Omicron booster. So now when you're exposed to Omicron, your antibodies are set up very well to take care of that Omicron variant that comes into you. So that pretty much wraps it up for me. I This again was a very biology heavy talk, but I wanted to break down this side of SARS-CoV-2 variants for you all because when new variants are in the news, we're always worried about immunity. We're always worried about if the vaccines are going to be good enough. And this is how we're making sure that's happening. And thank you all for watching.